environment is at this time, we really ignore soil carbon at our peril. So indirectly then, because fungi are really instrumental in regulating soil carbon, we ignore fungi at our peril as well. So I'll be talking a little bit about that today. I showed this last time as well, and fungi are the primary decomposers of plant biomass, and they have evolved a series of enzymes to break down the carbon-rich plant cell wall. So this is just a picture of the plant cell wall. You got in yellow, you have these are cellulose. This is a depiction of cellulose, and then you have these gray bits here, and that's lignin. If you look at the cellulose molecule, I don't know if you can see that, but it is this kind of long straight chain polymer, and it is these little glucose units that are here are linked by this oxygen bond, and that bond is called a glycosidic bond. It's not that important, but essentially, when it comes to decomposition, soil organisms are very good at breaking apart that particular bond because it's relatively easy to do so. But lignin, on the other hand, is a whole different story. And so lignin is again these gray bits in the plant cell wall, and it kind of confers structural rigidity to plants. It's the reason why plants stand tall or they can grow tall, because they have lignin in them. And this is what lignin looks like. If you compare it to cellulose, it's quite a amorphous molecule. So there's lots of these benzene rings, and that ring structure is really resistant to decay. So if you were a fungus, or an organism that wants to consume or get the carbon in plant biomass, it's much easier to decompose or to get it from cellulose than it is to get it from lignin. And so plants that have lots of lignin in them are by nature just more resistant to decay. But fungi are really good at breaking down lignin, and they're the most powerful decomposers of lignin on the planet. And then the way that they do this is they've evolved these really complex enzyme systems. And so these are enzymes. This is a really confusing figure, but these are all what's called peroxidases, and they are expressed by, or they released by mushrooms, and those things break down lignin and other plant biomolecules in soils. So I want to focus though on manganese peroxidase because it's arguably the most potent decomposer or enzyme that decomposes lignin. So let's just drill down into this for a minute. So I'm not really going to go too much into the redox chemistry here, but there's a couple of takeaways that I'd like you to have take away from this figure, and it's essentially manganese peroxidase, which is pictured here. It essentially turns manganese in the soil into from manganese 2+ into manganese 3+, and it takes away an electron, and then this manganese 3+ acts as this really powerful oxidant. And what that means is that it can break down lignin pretty efficiently, and it can attacks lignin in a non-specific way, whereas cellulose enzymes that break down cellulose are really targeting that glycosidic bond I was talking about. Whereas with manganese peroxidase, it kind of just, it's like a sledgehammer. It's essentially it just blows everything apart. And the end result of this is the mushroom gets carbon dioxide, or in some ways it moves lignin out of the way to get cellulose, and then it uses that carbon for its biomass. Okay, so yeah, some fungi have this really great enzyme, but not all fungi have this enzyme. And so I just want to go characterize the different groups of fungi to get you a flavor of how the ability to decompose lignin and other complex biomolecules varies within the decomposer community. So here we have, many of you are probably familiar with this, the Mario mushroom or Amanita muscaria. That's a ectomycorrhizal fungus that grows prolifically on the coast. It's what's known as an ectomycorrhizal fungus, and it gets its kind of carbon from tree roots. The trees actually give the fungus carbon, and it's sort of like a free lunch, at least relative to decomposers, to saprophytic fungi, which we'll talk about in a second. But the exchange, the tree gets nutrients, and the fungus gets carbon. And because of that, over the course of evolutionary history, these types of fungi have actually lost the capacity to break down carbon with manganese peroxidase explicitly. It's the kind of thing over the course of evolution, if you don't use it, you lose it, type of thing. So, but there are some genera like Fortnarius and Hebeloma that do retain this particular manganese peroxidase gene. Not very good decomposers, but let's talk about the really good decomposers. And so many of you are probably familiar with saprotrophic fungi, and you've probably heard things, fungi like white rot and brown rot. You've probably seen them in the forest, I'm sure, as well. So white rot fungi, like this guy, Fomes momentarius, they can degrade all components of the plant cell wall, including lignin, and these are the fungi that express manganese peroxidase. To contrast that with brown rot fungi, these are fungi that primarily break down cellulose. And then there's this group of fungi known as undefined saprotrophs. And these fungi, we don't know anything about them. We know very little about them, I should say. We don't know the kinds of enzymes that they have. But we know that they're doing something. We just don't know exactly what they're doing. So the point here is that the composition of fungal communities really does matter, especially when it comes to carbon cycling. Because, if you have, say, more ectomycorrhizal fungi, you have different types of saprotrophic fungi that break down carbon more efficiently, then that might reflect in soil carbon, the amount of soil carbon that's in the soil, yeah. Okay, so let's so from a fungal ecology perspective, the composition of fungal communities matters because different groups of fungi have different abilities to break down carbon. But let's pivot for a moment to soil geochemistry. And let's have a look at soil carbon storage across a large scale across Sweden, for example. This is a study that was released in 2017, and there's a range of different sites that the Swedes they have this amazing. They have 20,000 soil inventory plots across the country, and they visited 2,300 of them to try to see whether or not over this range of climate across Sweden, what were the primary controls on soil carbon. And so across these plots, the Swedes confirmed that manganese was the single most important predictor of soil carbon storage across all these plots. There were many other variables that were included in the model, from growing degree days to mean annual precipitation, other soil nutrients, tree species, and the amount of variation in soil carbon that was explained by these variables was 44%, and manganese explained 26% of that variation. So it really is an incredible predictor for soil carbon. And so the figure on the right here shows that relationship. And the relationship is when you have high amounts of manganese in the soil, you end up having low soil carbon. And so that kind of corresponds to keep that in the back of your mind, and then think about manganese peroxidase as well, because that's going to come into play in a second. But first, we're going to get to this work. It's moving a little closer to home on Vancouver Island. Marty, my colleague, has been working across a number of sites on Vancouver Island, mostly looking at phosphorus and nitrogen constraints on tree growth and the role that fungi plays in these kind of dynamics. And so he's been looking at these old multi-species spacing trials established in the early 1960s. And so there's all this different species, but we're going to be only focusing on Douglas fir. And there's just a really nice suite of 60-year-old Douglas fir plantations that were all planted at the same time. And they were planted across this orographic gradient. And so on the wet coast of Vancouver Island, you have podzols, and on the east coast, you have brunosols. And it's really because there's a very strong climate gradient here. So let's drill down into that a little bit. So on the wet west coast of Vancouver Island, the rainfall can be really high, as if anyone has visited in November, you probably know it rains a lot. They can get 3.5 meters of rain can fall in a year in some of these sites. And the site index, or the amount, or how tall a tree grows at 50 years, is very high. It's at 38.2. So just to compare to areas around Williams Lake, it's around 20 is kind of where things tap out around here. So in these systems where you have very high rainfall, you get these soil types to develop called podzols. These are really highly weathered relative to other soil types of soil. And there's lots of soil minerals like iron, aluminum, and manganese that are leached from the top of the profile deeper down into the profile. So that's just the nature of podzels. On the other side of the island, where it's drier, you have less than half the rainfall. You have these soils called brunissols. They are the rainfall is only 1,100 to 1,600 millimeters. And brunissols are, again, the main soil type here, and they're really less well weathered comparatively to the west coast. But the site index is relatively similar. It's 37.4 meters. So they're highly productive sites despite there being half as much rain. So Marty wanted to see if the trends that the Swedes reported could be replicated across his similar rainfall gradient that he's got on Vancouver Island at these sites. So the figure on the left is very nicely matches what the Swedes are finding. Wherever there is high manganese, there is correspondingly low soil carbon. It's just this nice relationship that kind of matches what the Swedes are finding. And through Marty's, he's been collecting mushrooms on the coast for a long time, and he has a really good feel for the types of fungi that make up the decomposer community across the range of his sites. And what he noticed was that there were some genera that were more abundant in eastern Vancouver Island on the drier sites that weren't really there on western Vancouver Island. In particular, genera such as Pseudohydnum and Clitosophy, they were more abundant on brunissols where carbon was low, but were very rare on the west coast under podzels. So what he wanted to do was test to see whether or not saprotrophs or ectomycorrhizal fungi, like how they respond to manganese content. He had this hunch that this is all related to manganese levels. And so this would give if saprotrophic fungi respond to manganese or are changing in manganese content over the soil carbon gradient, that it gives an indication that those fungi are actually somehow involved with manganese and soil carbon. And that's kind of what this graph is showing here. He took a bunch of mushrooms, and he found that over the soil carbon gradient, the saprotrophic fungi are responding by increasing their manganese content in the mushrooms, wherever manganese content is high. But ectomycorrhizal fungi depicted here, they don't really have that trend. And so what he gleaned from this is that the saprotrophs, because they have these manganese peroxidase genes, they're responding to the gradient in manganese over the soil carbon gradient. Okay, so now getting to the questions. The first question was whether or not brunissols had a unique fungal community with a functionally distinct potential for soil carbon decomposition. We know that certain taxa of fungi have more of this manganese peroxidase, and we need to demonstrate this across the sites. And then the other question, though, is okay, they might be differences in community, but what is that? Is there differences in function? Is there actually more manganese peroxidase expression that's occurring, or activity that's occurring in brunissols versus podzels? So that's kind of the first question. And the next question we wanted to look at is basically, can we modify the manganese availability to store more carbon in forest soils, which is a bit of a difficult question to answer, but effectively we wanted to see if we could turn podzels into brunissols by adding manganese. I'll get into that in a bit more detail, but first I'm going to tackle the first question. Okay, so we visited 10 sites in total, 5 podzels and 5 brunissols across western and eastern Vancouver Island, like in the figures I showed you earlier. Marty, Jill and I, Jill worked for me in 2019, and we all collected some samples in 2019. And there's Marty there. It wasn't a very good photo of his face, but there he is looking at a mushroom. So at each of these sites, we collected we collected the forest floor and mineral soil samples, and then Jill and I brought the samples back to Williams Lake and we extracted the DNA. Actually, Frank did a lot of the DNA extraction, and then we sent the samples away out for sequencing. We also did we did these things called enzyme assays, which is just basically measuring the amount of manganese peroxidase activity that's in these soils. We're quite happy in this photo because we had just been working out in 100 Mile. We have another carbon project out in 100 Mile, and in 2019, it was a wet spring, and it was just like so buggy. I don't know if you've ever worked out if you've worked out in the field in the spring in this region. It's just a real nightmare when it comes to the bugs. And we always joke that we had this luxurious fieldwork vacation when we went into the coast, because somehow there's no bugs on the coast, and it was quite a treat to be working on some of these sites relative to what we were dealing with, digging soil pits and mosquito-infested caribou. Anyway, that's an aside. But all right, so I want to give you a little flavor about how the soil differs between these two in chemical properties between these two types of soil, podsols versus brunosols. And the first thing that you'll notice is that brunosols, this is the forest floor in the mineral soil here. We've got brunosols on top and then podsols. And what's very common for podsols is you have high aluminum and iron, which is just a natural feature of these soils. And then manganese. We think that manganese gets leached further down the profile, and we're not quite sure why. But the point is that it's not picked up as an exchangeable form in podsols versus brunosols. And some other things. There was no difference in soil texture. Of course, foliar manganese, or the manganese content in leaves or needles declined by 60% in podsols versus brunosols, which is reflective of just the difference in manganese content between these soils. It's much higher in brunosols versus podsols. There's no difference in site index, as I was alluding to earlier, so or foliar N, or needle, the amount of nitrogen in needles. And so these are just indications that these sites are equally productive. So there's no differences in productivity, and there's no differences in disturbance history as well. And so these are effectively very similar sites, except for the fact there's a lot of rain in the podsols versus the brunosols. Okay, so this is an ordination, which I'm a little bit embarrassed to try to show you guys, because they're kind of hard to wrap your head around. But I'll walk you through it. But I glazed over the DNA sequencing part, but essentially what we can do nowadays is we can sample soils and many other substrates. We can extract the DNA and use sequencing technology to identify the types of fungi that are found within the sample. And so we can also tell how much what the relative abundance is of some fungi versus the others. We can tell how many fungi there are of that particular type. And so this technology has really revolutionized our understanding of microbial communities over it's really only been the last decade. But I want to stress, especially for this group, that it doesn't really replace naturalist knowledge. And this is reflected in this project, especially because Marty, one of the reasons that Marty initiated this study is because he noticed differences in mushrooms themselves. And so he's a naturalist, and he was able to see these differences, and then that's what got him thinking. So we can't just bury our heads in the lab and forget about actually picking up and looking at mushrooms, which I'm trying to do as I move through my career. But anyway, that's a bit of an aside. So I wanted to so this figure is what's called an ordination. The way you read them is that points in space that are close together have a similar fungal community, and so the fungi are just much more similar when the points are physically closer together in space. And so, looking at this particular figure, we have in orange we have brunosols, and in green, so the brunosols over here and the podsols are over here. And we have the two sample types. We have the forest floor in circles, and in triangles we have mineral soil. And so, like I said, with things that are close together are more similar to one another in their fungal community. And so you can see that all of the brunosol forest floor samples have a similar fungal community. All of the brunosols together have a more similar fungal community relative to the podsols. And so that's really what this figure is showing, is that the communities are very different. And that might be underwhelming, but having done this a number of times, this is a really exciting figure because the amount of variation that is explained by the soil type is very high. It's 25%. In some work it was like 4% or something like that. So this is a very good result in terms of explaining variation. And again, forest floor is also very different from, it's different from mineral soil as well. Okay, so it's not enough to really know that the fungi are different or the communities are different. We want to know exactly which fungi are most associated with one soil type versus the other. And so this is how we did this. This is a regression plot, and this gives you a flavor of the specific taxa that were different between the two sites. So positive values indicated in this side of the graph. These are Brunissol-associated soils, Brunissol-associated taxa, and then the negative values are Podzol-associated taxa, and that's just, again, these same colors, green and orange. So we had some ideas about which taxa were the ones that were really, would be different between the two sites. And so the fungi within this order called the Geochemistry, we think that they're the ones that have this manganese peroxidase gene, or that we know of that have the manganese peroxidase gene. And we were expecting in Brunissols that especially that a couple of saprotropic fungi would be within what I'm calling here as other agaricales would be more abundant in Brunissols, and that was the case. We had species like Cotopathie, Lepiota, Gymnopus, and Pseudohydnum. These were all more abundant in Brunissols. These are all saprotropic fungi. It's not clear whether or not they have the manganese peroxidase genes because that work hasn't been done yet, but at least they're going in the direction that we want them to. Then there's other groups, Stephoriaceae. These are Stephoria and of interest. Then Mycena and Clavariaceae. These are the coral fungi. These were more abundant under Podzols. Then there's these groups down here. These are just orders, which is a higher or lower resolution taxonomic rank, and it just means that there's more species in them. And so this group, especially, we know very little about. It's called the Trichosporales, and species within this group, which I'm not going to try to say, but they're like the jelly fungi. We found them to be more abundant as well in the Brunissols. What not showing here are species like or genera like Cortinarius, Piloderma, and Hebeloma. These are all ectomycorrhizal fungi that are known to have this manganese peroxidase gene. They're more abundant under Brunissols. So things are really falling into place, at least from what we were expecting. But is there a difference in terms of function between these two soils? And that's what this next part of the question is. And so these are the manganese peroxidase results. On the left here, we have Brunissols, and on the right, we have Podzols. In the light blue, it's the mineral soil, and in the polka dot blue, dark blue, is the forest floor. And then this is actually just measuring manganese peroxidase activity. What you can see is that the amount of manganese peroxidase that is occurring in Brunissols is 3.6 times higher than what is in Podzols. And so these soils have a certain group of fungi that are really pumping out this manganese peroxidase, whereas in Podzols, that's not the case. So to wrap up, to summarize this question, do Brunissols have a unique fungal community with a functionally distinct potential for soil carbon decomposition? And the answer to that question is yes. We showed strong differences between Brunissols and Podzols, and certain taxa were really coming out as different. Podzols, Brunissols also had a much higher activity in manganese peroxidase expression than versus Podzols. So that ticks those boxes. So that's kind of, we just kind of needed to prove that. But the next kind of almost more interesting question is that can we actually modify manganese availability to store more carbon in forest soils, given what we know about how manganese is important in regulating manganese peroxidase activity? So our approach here was to use a soil incubation study. So we just put some soil in some jars, basically some mason jars, and we modified the amount of manganese that was in the soils by doubling it essentially, or actually we took the amount of manganese and added as much as what would be in Brunesols to the podzol. And then we also added this inoculum of Brunesol inoculum to try to see if we add fungi that were from Brunesols to podzols, could we turn podzols into Brunesols that way? And so this is the basic design. So for each of the 10 plots and for each substrate, forest floor and mineral soil, we took soils and treated them with combinations of manganese or this inoculum of Brunesol fungi. So this is the control treatment here. It is we just basically added water in place of manganese and in place of the Brunesol inoculum. To isolate the effect of the Brunesol inoculum alone, we only added the Brunesol inoculum, and then we didn't add a manganese. And then to isolate the effect of manganese alone, we added manganese and not the Brunesol inoculum. And then finally, to see if there was a synergistic effect between manganese and the Brunesol inoculum, we put them both in. And we were kind of expecting things to happen along this trajectory. In podzols, there would be this increase in manganese peroxidase activity. The more manganese and the more soil inoculum or Brunesol inoculum you added, and this is kind of what it would look like. Whereas Brunesols would just kind of be flat. They already have the Brunesol fungi in there, and they already have enough manganese, so we wouldn't really expect to see results at all. What happened? Well, it was a bit of a different story. At first, it was a bit dejecting, but then we realized how interesting these results actually are. And so the manganese peroxidase activity for the forest floor is depicted here, and in this mineral soil in this graph, and we have the Brunesols and podzols. And so there's two treatments here. It's just talking about the manganese amendment. And so we have the control treatment in light blue, and then we have the manganese treatment. So we added manganese in this treatment in this polka dot blue color here. So what you can see is that when you add manganese to Brunesol soils, the manganese peroxidase activity declines by 56% after manganese amendment, which is very surprising. A similar decline occurred, but not quite as dramatic in podzols. So this is only for the forest floor. For mineral soils, the same thing happened, not quite as dramatic though. Add manganese to Brunesols, you see a decline in manganese peroxidase activity. But for podzols, in the mineral soil, things kind of went in at least the direction we were expecting. Increased in manganese peroxidase activity. So adding more manganese to podzols turns podzols into Brunesols when it comes to their manganese peroxidase activity. So this is a real conundrum, but the results are interesting nonetheless, and I'll tell you why. So to wrap up this question, Brunesol mineral soil and forest floors showed negative manganese response to manganese amendment. Again, this is not what we were expecting to see, and the reasons aren't exactly clear. But there's this body of research that indicates that the concentration of manganese actually regulates the expression of manganese in the fungi, and so it controls transcription of these genes. The reasons aren't super clear right now, and that's more work needs to be done there. But the more important point is that there's an inhibitory response when you add high levels of manganese. And for podzols, we found that manganese amendment actually increased manganese peroxidase activity as we expected, but there was a similar negative response for forest floors. And so what this is kind of showing us is that manganese amendment might be dose dependent. It's you just have to basically carefully add as much as that based off of the manganese content in these substrates to get the response that you want. Okay, so what does that all mean? We were working in our soil jars, but let's scale that up for a moment. And the good news is that manganese activity in Brunesols appears to be reduced by simply adding manganese. So if we can demonstrate that we can increase soil carbon, or we see a decrease in respiration, soil respiration in response to manganese amendment, we could potentially use this as a tool to store more carbon in forest soils. Originally, we were trying to do this thing where we were trying to somehow turn manganese 2+ into 3+, and that was technically more challenging, but it turns out that you can just add a bunch of manganese to these soils, and to brunosols, and manganese peroxidase activity really declines. So that's much less technically challenging task. So let's think about this on the scale of all the soil orders in Canada. You can see different types of soils in BC. You have this big plain of luvisols in central British Columbia. These are more similar they're similar to brunosols, and they likely have high manganese content and would potentially benefit from manganese amendments if you care about soil carbon storage. And then it was also brunosols all throughout Canada, and luvisols as well, which I think are two soil types that could benefit from this. So, like I said at the beginning of the talk, soil carbon is very stable, and so even small changes in soil organic carbon can have really big. This is just a picture of the River Valley because there's brunosols and luvisols there, but it's not really that important. Okay, so what does that mean for the future? There's a lot more work that we got to do, but one of the first things that Marty and I are doing once we get this paper out is to see if we can actually do this manganese amendment in the field. Can we increase soil carbon by adding manganese to forest soils? And then, we want to know whether or not these patterns extend beyond Vancouver Island, especially in this region where, in the east you have a lot of podzols, and in the Caribou Mountains, and here in town here you have a lot more brunosols and luvisols. So we can replicate the experiment, and then I kind of want to get at this idea of what's the appropriate dose to maximize soil carbon gains. And then in order to answer that question, we have to know things about, which are the taxa, which fungi are expressing manganese peroxidase, and then, what are the fundamental mechanisms that are control this dose response? And that's something that we don't understand yet. Excuse me, sorry. And then, of course, the big question is for people, like, what we really should be doing is stop burning so many fossil fuels, and that would be our solution. But we don't want to change, apparently. So the unintended, can we just add all this manganese and fix our problem by storing more carbon in soils? But I guess that might come with some unintended consequences. It's a very strange thing to have a soil scientist try to slow down decomposition, because decomposition is what releases nutrients and makes forests potentially more productive. So there could be some unintended consequences for productivity, although that isn't necessarily the case, at least on Vancouver Island. Okay, I think I've blabbed on enough, and yeah, that's pretty much all I got. I do want to thank some funders, of course, my employer, the Ministry of Forests, etc. The Capital Region District in Victoria allowed access to the plots. There's lots of field and lab support from Kate, Jill, Nay, Kathy, and Yulia, and then of course the lab analysis was done at the government labs. So thank you. We can take some questions.